Hi everyone, welcome back. This is the third installment of our lecture on digestion. And now we just talked about the gastric juices that get secreted in the form of hydrochloric acid and pepsinogen, which the hydrochloric acid then helps to activate to become pepsin. And pepsin is the enzyme that digests protein in the stomach. So we get major digestion of protein occurring. I did want to mention that when we swallow that bolus from the mouth, remember we have the enzyme amylase in there and it's breaking down carbohydrates. Well, the digestion of carbohydrates stops in the stomach because of the hydrochloric acid. The stomach environment becomes a pH of 2, whereas in the mouth, it's just a little bit on the basic side of neutral. It's just above 7. It's like 7.2 or something. And so that's where, so by the time the bolus hits the digestive juices in the stomach and it hits that pH of 2, then that denatures the amylase. So we stop digesting carbohydrates, but we do begin digesting protein. Okay, so how do we control this secretion, right? We want to make sure that we are secreting things at the proper time and we don't want to over secrete because we're using a hydrochloric acid here. So we want to be careful about that. So we have factors that stimulate gastric secretion even before food reaches the stomach. That's going to occur in the cephalic stage. Then we have factors resulting from the food in the stomach. So when food is in the stomach, that's going to stimulate secretion. And then when we're done in the stomach, we want to inhibit those secretions, right? So that's where the intestinal phase. This is going to be an inhibitory phase on the stomach. So the intestines will be functioning in a different way, but it's going to then try to shut off the gastric juices that's happening in the stomach. Okay, so let me give you just a little bit more detail on these things. So cephalic phase, the cephalic refers to your head region, right? So encephalization, it means a head is forming. So the cephalic phase is what's occurring in response to stimuli that are acting in the head before the food even reaches your stomach. So this is an example that we've used before regarding feed-forward processes, right? So maybe you're just thinking about food, maybe you're smelling it, maybe you're seeing it, right? Maybe you're even chewing it now and you're swallowing it. All of these things are occurring in the cephalic stage. And so all of these things then can stimulate your vagus nerve that stimulates the nerves that are infused throughout your stomach. That's called the intrinsic plexus. We'll see a little bit of that later. And that stimulates this production. It stimulates your parietal cells to make hydrochloric acid. It stimulates your chief cells to start making pepsinogen and pump that out so that you can speed up the digestive process just that much faster, right? You saw how long our digestive system is. So if we can just speed it up a little bit faster, have all the digestive enzymes ready to go by the time the food hits there, even better. Then when food actually enters the stomach, that's the gastric phase, right? And usually it's the presence of protein that stimulates the gastric secretion. So just the presence of protein being there will stimulate parietal cells and chief cells, okay? Then as we're in the stomach, then as we're ready, we're finishing the digestive process in the stomach and we're ready to put that, now it's a very liquidy substance called chyme, and that's what's written here for you. So the chyme is now a mixture of the gastric juices and some of the broken down foodstuffs even more, whereas before the bolus was kind of almost a solid but mushy substance, whereas now the chyme is liquidy. And as that then enters into the small intestines, we start an inhibitory phase. It helps to shut off the flow of gastric juices so that we can end what we're doing in the stomach and now continue what's going on in the intestines. So that's what you see here. Okay, so the food came in, our gastric juices are in here. So we not only get just the juices laying here, remember I mentioned before, we have mixing occurring. So you have smooth muscle layers, especially down in the antrum where you have thick muscle layers. You get these peristaltic waves of contractions that originate in that upper fundus here. Remember, we had some pace setter cells that can, the intrinsic plexus is infused throughout the muscle cells here to stimulate them. And so you get these waves of contraction, which kind of like squishes the stomach all the way down and squishes the food. Let me do this picture first, up against the pyloric sphincter, which is closed at this point, right? And so that squishing of the food down causes the chyme then to hit the sphincter and bounces it backwards. So you get this mixing back and forth, back and forth, back and forth. Okay, and it just tosses the mixture. So it's mixing the food and it's getting the gastric juices mixing in there. And so now the enzyme can hit those proteins and really get that mixture going, right? And getting the digestion going, right? And so that's just going back and forth, back and forth, back and forth. Okay, then as it gets really thin, right? Sorry, I was just making sure I wasn't missing something here. Yeah, so how does the stomach know when it's ready to empty, which is this picture that you see here? Okay, how does the sphincter know when to empty? Okay, it's largely controlled by factors in the duodenum here. Okay, so when the mixture in here starts to get really thin and becomes that chyme, a small portion of the chyme then actually squeezes through the pyloric sphincter and gets into the duodenum. Okay, and so then as this acid environment, or as the environment of the duodenum starts to go down in pH, meaning increases in acidity, that's then going to send signals to stop the actions of the stomach, right? That's back to that intestinal phase. All righty. I think what I was looking at, I did want to mention that just some properties of the stomach itself before we go into this emptying. So when your stomach is actually empty, its volume is only about 50 milliliters, but then it expands to a liter in a meal. Okay, so from 50 milliliters to 1 liter during a meal. And again, but the pressure inside doesn't change very much, and that's because of that rugae that I had mentioned before. So as you're filling your stomach, the rugae start to expand first. And so there's very little pressure being pressed against the mechanoreceptors inside of there, right? And so again, that just enhances the stomach's ability to accommodate this extra volume of food with little rise in the stomach pressure. Okay. And so if there's more than a liter of food, the stomach becomes over distended and this pressure inside the stomach arises, and then you start to feel discomfort. All righty. So let's look at now, we just said as the chyme now is becoming more and more liquidy, the next time that it slams against that pyloric sphincter, it actually squirts a little bit out. And so now we get some into the duodenum. So what's going to promote this, what's called gastric emptying, right? Releasing the chyme out of the stomach. Well, one of it is factors in the stomach itself. The amount of chyme in the stomach is actually the main factor that influences the strength of a contraction that's occurring, right? All that peristaltic wave, right? And so it'll start to even contract even harder, which is going to put pressure against that sphincter, causing it to open just a little bit, right? Then then we get into factors in the duodenum, right? As factors in the duodenum, as fat starts, so it says it inhibits gastric emptying, right? So basically what's happening, the amount of chyme in the stomach causes that sphincter to open, right? And it does it in little squirts, right? And so this is important in the next phase here to inhibit the gastric emptying. So it does it in little squirts. So as a little squirt of chyme comes out, that means the acidity lowers, right? Or I should say the acidity increases, the pH lowers in the duodenum, the first part of the small intestine, a little bit, right? And then there's some fat in there now. All of a sudden, the duodenum can sense that there's fat inside of there. Because remember, we really haven't had any significant fat digestion in the mouth and in the stomach. Okay, so this is starting to rise in there. Okay, and so as this unneutralized acid begins to fill up, what that means is more and more chyme is filling in there. As the fat content increases, that means there's more chyme entering, right? As the duodenum becomes more hypertonic, that means more chyme is entering, okay? As the duodenum starts to get stretched, that means more chyme is entering. So all of these factors are signaling, hey, we've got more and more chyme now in the duodenum. That must mean there's less in the stomach. So let's slow down the activity in the stomach. Okay, let's slow down what's going on there. So now we're going to start to get feedback loops back to the stomach to slow down the parietal cells, slow down the chief cells. We don't need to be producing acid and enzymes anymore in there. It's going to inhibit the peristaltic contractions, right? So again, I'll say it again. To promote the gastric emptying is the increase in chyme in the stomach, right? What then begins to inhibit the gastric emptying more and more and more is as the chyme begins to increase in the duodenum. Okay, and we detect that in the duodenum in these 4 ways. One is just the duodenum getting wider because it's filling with chyme, but also chyme is acidic, so we can sense the acidity. Okay, we can sense the amount of fat now that's coming in. Okay, we can sense the amount of other molecules in there because it's becoming hypertonic. All of these things are signals that the stomach is emptying out and it's coming into the duodenum. We want to stop the activity of the stomach. So we'll start to inhibit that. So that's the next slide here. What triggers, so again, the triggers for gastric emptying and what slows it down. Okay, so we have a neuronal response, right? You can, again, that nervous system plexus that fuses through our stomach, it can be separate from the brain, right? It can be separate from the autonomic nervous system. It's what's called a short reflex, okay? So that's the intrinsic nerve plexus, okay? But it also is controlled by the autonomic nerves. That's called a long reflex, right? So this is the that enteric system that we saw on the neuron, the flow chart that shows the structure of the nervous system. And we saw the autonomic nervous system and the sympathetic and parasympathetic. And parasympathetic controlled the stomach or controlled the digestive system. But then there was that offshoot, the enteric system. That's this intrinsic nerve plexus, so that it can kind of control itself. It has its own reflexes in there. Okay. There's also a hormonal response that can occur, right? And so remember this other slide. Let me go back a little bit. The one that had the table, which is right here, right? So remember there are endocrine and paracrine cells that release hormones. So the G cells release a hormone gastrin, and the D cells release one somatostatin, right? And so these help to control. Let me go back to this slide. There it is. So there are those and more hormones listed here for you. Okay, so in the hormonal response, this involves the release of hormones from the duodenal mucosa. Okay, when we're down here with cholecystokinin and the GIP and secretin. Okay, and I guess all of them. I guess gastrin in there as well. No, gastrin is released by G cells in the stomach, right? So secretin, cholecystokinin, these are from the duodenum, okay? But let's go through each one first before I confuse you on this. So gastrin, again, its release is stimulated by the presence of the protein in the stomach. Okay, so this one is going to want to promote the digestive activity in the stomach. So if the stomach receives a signal that protein is rising within the stomach, remember the stomach has to digest protein. So a hormone gets released out of the G cells within the gastric pit, which is going to stimulate our parietal cells and our chief cells. Right, so those release hydrochloric acid and pepsinogen. Those are going to promote the digestion of the protein in the stomach, right? But then as the chyme then gets released into the duodenum then, the acid in the duodenum stimulates the release of secretin. Right, so secretin now is going to inhibit gastric emptying in order to prevent further acid from entering the duodenum until the acid that's already present there from the chyme entering gets neutralized. Okay, so it's going to inhibit the gastric secretion to reduce the amount of acid being produced. Okay, but secretin sounds like secrete. So it's also going to stimulate the pancreas to release large amounts of sodium bicarbonate. Sodium bicarbonate is also a buffer. It can reduce the acidity. Okay, and it can also cause the signal the liver to also secrete bile, which is rich in sodium bicarbonate as well. Okay. So again, secretin is going to inhibit the gastric secretion, right? Because it's sensing that acid is increasing too much in the duodenum. So we want to slow down the release of chyme. That's going to slow down the acidity. But we're also then going to get two other organs to secrete a buffer. That comes from the pancreas in the form of sodium bicarbonate, and also in the liver in the bile that gets released, the sodium bicarbonate. So those two things help to neutralize the acid. Okay. Then we have this one, cholecystokinin. That one's fun to say when you figure out how to say it, but it's also called CCK. Right. So cholecystokinin also inhibits gastric motility and the secretion. So that's going to be a negative effect on the stomach to prevent that contraction of the stomach, which is going to prevent more gastric emptying, and it's going to prevent the secretions again. It's going to prevent the hydrochloric acid and all that stuff. So we're doing things now, again, the triggers in the duodenum are causing the release of these hormones now to slow down the activity in the stomach. But cholecystokinin can also stimulate the pancreas to start to release its pancreatic enzymes, which we're going to get into in a minute. Okay, and so remember now, through this process, we started digestion in the mouth, but we only started with carbohydrates. We didn't finish because it stopped when we hit the stomach. In the stomach, then we started digesting proteins, but proteins are difficult to digest, so we don't finish with that either. Now that this chyme is entering the stomach, we're going to neutralize the acid, and we're going to get the release of more enzymes in order to continue and complete the digestion of carbohydrates, proteins, and now lipids as well. So all of that's going to happen in the small intestine, okay, in the duodenum, right? And so these hormones are what's controlling. Let's stop the activity of the stomach, and now we're going to stimulate the activity in the duodenum, and we need enzymes coming from the pancreas, right? So cholecystokinin, we said, helps to stimulate the secretion of those enzymes coming from the pancreas. Okay, we also get some secretions from the gallbladder, which is near the liver, right? And so it's going to, cholecystokinin is going to trigger the contraction of the gallbladder so that it squirts the bile into the duodenum as well. Okay, and then GIP is a hormone. It stands for glucose-dependent insulinotropic peptide. You can call it GIP. What it does is it's starting to, because pretty soon we're going to start absorbing all of this food, which is going to contain a lot of glucose. So GIP stimulates the pancreas to release insulin. And remember, insulin stimulates all our other cells to pull glucose out of the blood. Okay? Whew, that's a lot of control, right? You got to get these hormones straight, right? And like I said, there's some tables in the book I think can help you out. But what else? There are other factors that can affect your digestion, right? Emotions. If you're sad, fear, that tends to slow down the activity of the stomach, right? You don't want to eat very much. So emotions can do that. Anger, aggression, that tends to kind of increase the motility. So sometimes you hear the stomach growling, right? And so that can do that. So again, that's all coming from the cerebrum. Intense pain, that tends to inhibit motility. All righty. Okay. Now we've got chyme, right? This picture entering our duodenum. As it's entering, we've got hormones that are inhibiting this activity in the stomach, but it's stimulating the pancreas and the liver. So now let's go into the duodenum, see what happens there. There it is. Here's the duodenum. The duodenum is part of the small intestines, which consists of itself, the duodenum, the jejunum, and the ileum. Okay, and so they're all connected to one another. They're all one organ, okay, but they just have different functional units, right? The inner lining, the mucosal layer, will differentiate a little bit, right? But the duodenum is the site of most of the digestion. So again, we saw a little bit in the mouth, we have some in the stomach, but the rest is going to occur and complete in the duodenum. It's only 10 inches long, and you can see a little segment of it because the rest of it is behind the large intestine here. The next segment is the jejunum. This is where most of the absorption of the nutrients will take place. And then the last segment is the ileum. This is 12 feet long. And this is where we'll get the rest of the nutrient absorption, but mostly water and electrolytes and things like that. Okay. Peyer's patches are more common. That's part of the lymphatic system, if you remember that from anatomy. But let's go back into the duodenum because we haven't completed digestion. We have to see what happens there with digestion. So I mentioned though that the duodenum, maybe I didn't mention this part, but the duodenum doesn't make its own enzymes for digestion. It receives them from the pancreas and from the liver and the gallbladder. So we need to look at that. So let's look at the pancreas, which lies just underneath the stomach. So if we pull the stomach up here, you'll see the pancreas lying there. And then the pancreas has a duct that goes down the middle of the pancreas. You see these branches that are coming from different cells here. So the acinar cells or acini cells are the exocrine cells. These are the cells that are producing the enzymes. And then also above in the duct, there are the cells that are producing sodium bicarbonate. Okay, so it's bicarbonate, but it's got a sodium attached. Okay, and all of that gets secreted down this duct, which connects to the duodenum. So that's how the duodenum receives the enzymes as well as the bicarbonate in there. And the bicarbonate, again, is really important in reducing the acid. Okay, so that's the exocrine function. Okay, so I'll show you in a minute. The pancreas makes several types of enzymes. So altogether, we just call them pancreatic enzymes. Okay, but then also it mixes with that alkaline sodium bicarbonate, and so it makes that aqueous solution. Okay, and so that's going to neutralize the pH in here. It's going to make it back down to like a pH of 8 or something like that. It gets pretty basic, but again, these enzymes then, they don't function well in the acid environment, so they need a basic environment. Okay, the endocrine function of the pancreas are these little things here. Remember the islets of Langerhans. So these are the cells that make and secrete insulin and glucagon. So I already had mentioned over here that GIP, right, signals the pancreas to start releasing insulin, because once digestion's completed here, we're going to start reabsorbing or absorbing all of that new glucose through the jejunum and into the bloodstream. So the bloodstream will get a spike in glucose. So if we can get insulin out there and get cells to start to take the glucose up right away, then we don't get as much of a spike of glucose, right? All that extra glucose can affect the tonicity inside of the blood and affect the absorption of water and things like that. So having a healthy supply of insulin helps to prevent all of that. Alrighty, so let's look at the exocrine secretion though, right? So again, the exocrine secretion of our pancreas is regulated by what I just mentioned, secretin and CCK, right? So secretin, we said, regulated the secretion of the sodium bicarbonate, and CCK stimulated the release of the enzymes. So all of these enzymes are pancreatic enzymes, also known as proteolytic enzymes. We've got to digest everything now. So we've got several that digest protein, and again, the pancreas is going to release an inactivated form called trypsinogen. And once it hits the juices inside of the duodenum, it gets converted to trypsin. So that's going to digest protein. There's chymotrypsinogen, which gets converted to chymotrypsin. Again, it's all going to help digest protein. Once protein, the bonds get broken, remember you've got to unravel it and break bonds. So there'll be different segments. So these different enzymes help to break them down into the individual amino acids that they are. Okay, and then there's pro-carboxypeptidase, and that becomes carboxypeptidase. Okay, and pro is another, instead of ogen, pro is another term that means it's inactive. Okay, I mentioned pancreatic amylase, right? So this is going to help break down our carbohydrates. So we still have polysaccharides, and we're going to help break them down into amylose, which is a disaccharide. Okay, and then we're going to get significant digestion of our lipids. Okay, so a little bit of digestion in the mouth and the stomach, nothing to write home about. But now the pancreas releases a pancreatic lipase. Okay, there's actually a lot of different names to them. If I'm just giving you lipase to make it easier. Okay, so this is the fat. You know what? This isn't quite true anymore. Only fat digesting enzyme, only significant fat digesting enzyme secreted throughout. Okay, only significant fat digesting enzyme. So all of these go into the duodenum, and again, starts digestion or continues digestion, keeps it going, right? But that's not the end of it. Remember, the luminal surface of the duodenum has all these finger-like projections, and it makes it look like a brush. Those are called that's called the brush border. So here's showing the cells, and you've got all these foldings of the luminal surface, right? That's called the brush border. So there are actually enzymes embedded within the brush border of the epithelial cells. Okay, so remember over here, I mentioned that, I keep triggering Siri, that we can break down into a disaccharide. Well, disaccharides can't cross the luminal surface. So these borders then have an enzyme embedded in the cell membrane that can then help break, this is a different type of disaccharide, but it can help break it down into the monosaccharide. So then when that moves into the jejunum, that it's ready then to take these monosaccharides in. Okay, so within the duodenum itself, again, there's enzymes embedded in the cell membrane, and those are called enterokinase, disaccaridases, and aminopeptidases, right? So that's going to help continue the digestion down into amino acids. Okay, was there something else? Yeah, so this continues, these three are carbohydrate and protein enzymes. The lipids fats are digested entirely within the lumen of the intestine by that pancreatic lipase. Right? All right, we're getting there. So that's the contribution of the pancreas to the duodenum, right? The liver also contributes as well. So here you see the liver kind of sits up above and on the right side of the body, where your stomach is on the left side of the body. So it's almost equal with the stomach, but a little bit over it as well. And so you have the liver inside of there. The liver creates what are called bile salts. It uses cholesterol to make bile salts. And then it stores the bile in this organ, the gallbladder, and that is embedded right up underneath the liver. And so they share a duct that then travels down to the duodenum. And so that's where the biles then get released into the duodenum. So remember, again, the liver keeps making bile and then it stores it in the gallbladder. And then when the chyme starts to enter the duodenum, remember that's the release of that CCK. And CCK causes the contraction of the gallbladder. It's like a little balloon and you just squeeze it and squirts bile down into the duodenum here. Okay, so it's the liver is really important organ, and it's only one small function is digestion. It's got a lot of other functions. Okay, but without the secretions of the bile from the liver and the gallbladder, the lipase wouldn't be able to work on the fats. The enzyme wouldn't be able to work. You need these bile salts and the bile itself to help kind of pull lipid molecules apart. So think about when you have oil and water and you mix it up and you see all the little small oil droplets, but then they fuse together and they form these big droplets, right? Well, those big droplets are too big for the enzyme to grab onto. So what bile does, let me go to the next picture here. Here are the little lipids, right? What bile does is it kind of separates the lipids from one another, keeping the droplets from merging together and forming a big droplet. It keeps them separated. So now the enzymes can in there and actually work on it. Okay, so that's the main function of the bile salts coming from the gallbladder and the liver for digestion. So again, without the bile, it's very difficult to digest fats because the enzymes can't work on it without it turning it into that structure called the micelle, keeping the droplets small. So people who get their gallbladder removed, if you do that, you've really got to reduce the amount of fats in your diet. Fats then just keep going through your intestines and you excrete it out of the body. Okay, so it helps to really reduce your fat intake if you get rid of your gallbladder. Okay, the liver, like I said, has a lot of functions not even related to digestion. And so in the circulatory system, when you might have learned this in anatomy, when the blood that goes by the stomach and also by the intestines, it'll get picked up by the hepatic portal vein. So all of the blood then that picked up the nutrients and absorptive surfaces from the small intestines, it'll all get taken to the liver first. And so it's in the liver that it can detoxify any of the substances in there. It can process further some of our nutrients. Some of them need further processing before our other cells can use them. Just kind of metabolizes toxins, breaks things down, so that then the blood that gets returned to the heart to get pumped out to the systemic system is nice and clean. So the liver is really important in doing that. Remember that it creates plasma proteins, albumins, globulins, all of those things. It's an important organ in storing glucose in the form of glycogen. Right? It stores fat, it stores iron, copper, vitamins, so really important. It activates vitamin D, okay? It removes bacteria, worn out red blood cells, right? And it can excrete extra cholesterol. And bilirubin is a breakdown product of red blood cells, right? So if it removes worn out red blood cells, the breakdown product of that is the bilirubin. Okay, so again, the liver has a lot of other functions, not just digestion. Alrighty, I'm going to end it there because then we will hit the rest of the small intestines and I believe finish it up in the last video. Alrighty, take a break.